active transport. So active transport is first is primary, second is secondary. So let's study first primary and then we'll jump to secondary. So primary is primary because it uses ATP. And how we can use ATP? We can use ATP with the help of ATPases. This channel has their own ATPases. These are like pumps for ATP. For example, these are like pumps and they use ATP for the transport. Basically, this is the concept. So one of the pump is sodium potassium ATPase. Another is H plus potassium ATPase, proton pump. Cool. It's hydrogen, by the way. So calcium ATPase. Cool. So the concept is that this there is transfer of ATP and substance can travel. Cool. That's the concept. So let's talk about sodium potassium ATPase. So sodium potassium ATPase is usually how it works. It has two subunits, alpha and beta, and this pump actually works in a cool way. It's how it works. It works three sodium out of, like, out, it's outside is extra membrane, inside is intramembrane. So three sodium outside and two potassium inside. So overall it's a little bit electronegative. Cool. It's overall a little bit electronegative. So alpha subunit, beta subunit. So it has two subunits. So that's why, and these both of these are like different subunits. So we call this heterogeneous heterodimer because both of the subunits are different. Cool. Alpha subunits binds to 5 binding sites, and beta subunits has 3 glycosylation sites. Now glycosylation means it's connecting to glucose at 3 sites, and alpha subunits is connecting to 5 sites. Cool. Let's see what are these 5 sites. So again, 3 out and 2 in. Okay, so alpha unit connects here. First is the outside and other one is inside. I'm talking about the sites. So there are 3 sites that are inside the cell and there are 2 sites that are outside the cell. So the sites that are inside the cells are potassium. Why potassium? Inside cell potassium is low, but some potassium still present and it's continuously being exported. It maintains the gradient. Cool. ATP binding site. Cool. Phosphorylation site. Cool. And outside there is potassium binding site, which is also important, and ubiane, which inhibits this pump as well. So these are like sites. Outside there are 2 sites, inside there are 3 sites. So there are 5 sites. Let's talk about the coupling rotation. Coupling rotation is like what's happening. So 3 sodium outside, 2 potassium moving in, and this is electrogenic pump. Cool, because it's making a negative. The net result is negative net result is negative electronegativity of the cell. But why to kick potassium? Why do you want to kick potassium? Cool. I also think this. Why do you want to kick potassium? Why you want to bring this? Why do you want to, not potassium, sodium? Why do you want to kick sodium? Why? Because inside the cell, the proteins and other anions like DNA, RNA are already negatively charged, and they cannot leave, because if they leave, then what is even cell? It's nothing. So if Na stays in, the water would rush in, because we already know Na, sodium, is osmoactive. It brings a lot of water. So we don't want our cells to swell up, right? So that's why we need to kick out Na. So water would rush in via osmosis and cell dies, swelling. Cool. And 3 Na out and 2 Na, it makes the cell more electronegative. Cool. And note, resting membrane potential is -70 and negative interior needed for, we need, like, so you get the idea, right? We have to do this. If just imagine if we had 2 inside and 2 outside, the net result will be 0. And we want cell to be a little bit negative. We want this resting membrane potential to be negative. We don't want this to be positive, because at positive, these DNA RNA will not sustain. So that's the idea why the sodium is kicked out and potassium is brought in. Cool. What are the function of potassium and sodium potassium pumps? Cool. What is the function? They regulate the cell volume. Okay, they regulate the cell volume by regulating sodium, because water follows sodium. Cool. They maintain the resting potential membrane. Cool. They maintain it by maintaining a overall negative charge. Cool. Indirect energy for secondary transport. I'm gonna tell you about this SCORA soon. Don't worry. Okay, how to regulate sodium potassium ATPase pump? We can regulate by increasing its activity or decreasing its activity. How to do it? We can decrease its activity by dopamine, ubiane, digitalis. Now, ubiane and digitalis. Ubiane is one of the site that is outside. It's an alpha binding site that's present outside. With another binding site is K, that is potassium. So potassium and ubiane are the binding sites that are outside the cells, and they are alpha binding sites. So ubiane and digitalis are quite similar, and dopamine as well. It reduces their activity. What about increasing it? Thyroid, insulin, aldosterone. Cool. Now aldosterone is notorious for NA. Cool. Remember this. Cool. Thyroid, insulin, and aldosterone. Thyroid is the growth works for growth. Insulin also works for growth, and aldosterone works for NA. Just remember this. Okay. NA, this sodium potassium pump increases equals what it does. It increases the ATP hydrolysis, and it increases the metabolism. What is ATP hydrolysis? It means that the ATP is breaking. ATP is breaking with H2O, and what it gives, that ATP plus 8 ATP, it gives ADP, okay, plus some phosphate, and this is how this happens, and plus energy. Cool. We use this energy. Cool. Let's go to secondary one. What is secondary? So that was primary active one. Let's go for the secondary active transport. So secondary active transport, it uses the energy gradient from the primary one. So that's why we call this secondary, because it's not primary, so it's secondary. It utilizes the energy from primary transport, as sodium is more outside, high concentration, and with that sodium glucose also come inside because of the concentration gradient. Just imagine, because of the sodium ATPase pump, a lot of sodium is outside the cell on extracellular space. So what happens is that a lot of sodium is outside, and concentration is up, a lot of sodium, and inside the cell there is very less. So nature wants to, we want to achieve equilibrium, we want to achieve nirvana. So how the cell achieves its nirvana is transporting some sodium inside back, and with this sodium some glucose as well comes with it as well, right? So that's the concept. So example, so these are, by the way, passive, these are active pumps, of course, but they work on concentration gradient. Okay, they work on the concept of concentration gradient. Cool. Example, sodium glucose link transport, SGLT. Now there can be SGLT1, SGLT2. SGLT1 found in gut because you have only one gut, and SGLT2 is found in kidney because you have two kidneys. That's how I remember it. Cool. Symporter. Symporter means if one is going out, another one is going out as well, because this is sim. It's together. What is exchanger? Antiporter. One is going out, another one's going in. That's exchanger or antiporter. Symporter, as both Na-glucose works in same direction, opposite direction of Na-glucose, we call it antiporter. Cool. Another example, sodium potassium 2 chlorine co-transporter. Now it's a co-transport. It means all of these things are getting out same direction. Where can we see them? We can see them thick ascending loop of Henle. Cool. What about sodium iodide transporter? We see them in thyroid gland because we want iodide to make Th3 T4. Cool. Another example, chlorine bicarbonate exchanger. It's an exchanger. It means the direction is opposite. So in this way, these are present at RBC, your red blood cells, and what happens is that chlorine comes in and bicarbonate jumps out. Now why this happens is that RBCs have CO2. Cool. Now this CO2, the carbon dioxide, gets inside the RBC, and this carbon dioxide converts into bicarb, and this bicarb has to go out of your body via lungs. Cool. So that's how bicarb pushes out RBC. It's called anion exchange because these are both anions. Cool. What about this different one? Chlorine out, bicarbon in. So this happens at lungs. Why it happens at lungs? Because we wanna exhale. So bicarb is in RBC already. So chlorine moves out and HCO3 can turn back into CO2. Now this thing happens in RBC and then we exhale. I hope you understand me. I don't know, I'm a terrible explainer at this thing, I think. Okay, cool. Just understand this, that sometime chlorine can get out and HCO3 can come in, and HCO3 can get out and chlorine can come in, or like Nava Road, or like opposite. Cool, now you understand. So we have vesicular transport as well. This is active as well. So what happens in vesicular transport is there can be exocytosis, can be endocytosis. Exo means outside, endo means in, cytosis means some the cell is producing. So exocytosis and endocytosis. What is it? Cool. Both are active transport. Important. Cool. They both need calcium. Very important. And where is calcium stored in the cell? First of all, calcium is not stored a lot inside the cell, and second of all, like, calcium is stored, of course, but it is stored in sarcoplasmic reticulum, and this calcium is responsible for the signaling, and that's how the vesicles get out. Okay, exocytosis and endocytosis. For both we need calcium, and the most important is that there is no role of concentration gradient, because vesicles are quite unique, and you don't have a lot of vesicles waiting outside the cell, because vesicles are important, so cell doesn't make them like in abundantly. Cool. If both endo and exo are happening simultaneously, like one vesicle getting out, one vesicle getting in, we call that transcytosis because it's across, and we can call this cytopempis. Cool name. Okay, let's try to study about exocytosis. What is exocytosis? Exocytosis is when the vesicles are getting outside the cell. There is a protein called SNARE. Now the SNARE protein name is quite complex. It's not complex, it's just a long name, so I didn't write it down. So this SNARE protein is present at the membrane. Just try to imagine this. And now there are two types of SNARE protein. One is synaptotagmin and another is synaptobrevin. Cool. One stands with tagmin and another one is brevin, and both are synapto. Cool. So just imagine this is the membrane, and here is one synaptotagmin and here is synaptobrevin. Okay. So this synaptotagmin senses calcium. Again, I told you calcium is needed for signaling. Okay. So once it senses calcium, what happens is that the vesicle is present at. Okay, so again I was wrong. Okay, I forgot. Cool, I can be wrong, I'm just a student. So this is a vesicle, and at this vesicle there's one protein here, there's another protein here, then there is here one protein, and then there is here one protein. So these proteins that are present on the vesicle are called synaptotagmin and synaptobrevin, and these proteins that are present on the membrane, we call them SNAP25 and synaptotaxin. These are present on the membrane. Cool. So what happens is that this SNAP25 and this another one, synaptotaxin, SNAP25, synaptotaxin, vesicle, Tagmin, brevin, they just connect with each other, and then the exocytosis of the vesicle happen. And this happens. This everything happens with the with signaling from calcium. Very important. And who can sense calcium? Tagmin. Okay, so that's the concept. If calcium is present, sensed by synaptotagmin, the exocytes occur, and these proteins SNAP25 and syntaxin are present on membrane. So that's the concept. What about botulinum toxin? So there's something called botulinum toxin. It's Botox basically, and it produces flaccid paralysis, and it produces flaccid paralysis because it stopped acetylcholine. So okay, I'm gonna tell you how. What happens is that this botulinum toxin, it destroys the brevin, synaptobrevin, and once it destroys the synaptobrevin, no exocytosis can happen. So flaccid paralysis, as acetylcholine is released in vesicles, but botulinum destroys the brevin, so no acetyl. If no acetyl, then no contraction of the muscle, so there is flaccid paralysis, because acetylcholine is responsible for the contraction and relaxation of a muscle. So that's why you don't want botulinum toxin, and that's why Botox works. Cool. What about endocytosis? Something getting inside. The vesicles are getting in. Cool. There can be three types of endocytosis. First is phagocytosis of the bacteria, and other one is pinocytosis of fluid-filled vesicles, and the most important one, I think, is receptor-mediated endocytosis. You need a receptor, and once the receptor is happy, there can be endocytosis of a vesicle. What is it? LDL is taken inside. How? Via receptor mediated endocytosis. It's done with the help of clathrin and dynamin. Okay, clathrin. Okay, these two proteins are needed to take LDL. Okay, inside. Okay, it's done via receptor mediated endocytosis. Okay, again, note, if a substance needs a channel, of course, or a carrier to transport itself, it takes some time to free up these channel or carrier, because they can be overloaded. There are not infinite number of channels and carrier in the world, in the cells in your body. They are limited. That's if the demand increases a lot, the saturation comes and the plateau hits. But a simple diffusion never hits saturation, because a simple diffusion is not carried by any channel or by any carrier. So this is the carrier channel, this is the simple diffusion. Now with time, simple diffusion keeps on walking, keeps on happening, but the carrier protein this carrier transport always hits a plateau. So that's the concept. Cool. Done with this active and passive transport. Let's talk about membrane. Okay, let's talk about membrane potential in the next one.